so the title of this lecture is Plant Hormones in Environmental Signaling. Okay, and as I was telling you, I would like you to get the idea in the end of the talk that the hormones are really important for the plant to interpret changes in the environment. Okay, so I cannot pass the slides. So how is okay now? Okay, so these two researchers, Fritz Wendt and Kenneth Thiemann, they wrote a seminal book almost 90 years ago called Phytohormones that it established the conceptual framework for all future research on hormone signaling. Okay, and in this book they describe hormones as a molecule that being produced in one organ are transported to another part of the plant where this molecule triggers a physiological response. Okay, and in this book they emphasized a lot the functional aspect of these hormones, of these molecules, saying that they are chemical messengers, okay, connecting different plants of the part and coordinating their activity. Okay, so although this is true for many hormones, and this view is a bit outdated right now, okay, because we already know that all cells in the plant are able to produce hormones, okay, and in many cases, these hormones can act in the very same cell where they are produced, okay, but in any case, the plant hormones term, it's still valid, okay, because they are produced and they act in very, very tiny amounts, and they have dedicated signaling pathways that are triggered by the hormone to provoke a physiological response. Okay, so these are all you know, these are the hormones in blue. Are painted or highlighted the classical hormones, and this is because they were first discovered, all of them during the first half of the last century. And I would say that auxin, the first experiments leading to the discovery of auxin were designed by Charles Darwin. So we have to go back to the 19th century. Okay, and hormones regulate virtually all aspects of plant physiology, okay, in one way or another. And particularly interesting for this talk is that, as I was mentioning this before, they are very important for the plant they help the plant to cope with environmental changes, okay, all over the life cycle of the plant, okay. All hormones, in one way or another, they are regulated by the environment, okay. In many cases, this regulation is through the regulation of genes encoding key enzymes in the metabolic pathway. Okay, and as I mentioned before, we will illustrate this effect of hormones interpreting environmental signaling with 3 examples later on. Okay, but before going on, I would like to open a parenthesis here to say that in some cases, hormones are integral parts of developmental pathways. Okay, and in these cases, they have to be buffered against environmental changes, okay? And we can illustrate this with a couple of examples, okay? So, as you can see here, the high cytokinin activity, the opposite cytokinin activity in this cell and auxin activity in this cell, these are globular embryos from Arabidopsis, okay? And you can see here how this cell has a high cytokinin activity visualized with this reporter for cytokinins, and this cell has high auxin activity visualized with this other reporter, okay? So this opposite accumulation of cytokinin and auxin, this is important to determine the identity of cells in the root, okay? So this apical cell with high cytokinin activity is the precursor of the quiescent center, that is the part of the root where are the stem cells, okay? This is very important. And this cell, can you see the pointer, right? And this cell with high auxin activity is the precursor of the columella, that it's also a special type of cells in the root, that it's very important. So it's reasonable to think that the activity of cytokinins and the activity of auxins in these cells are buffered against environmental changes that in other developmental contexts may affect the activity of this hormone, okay? Because the identity of the quiescent center and the activity of the columella cells have to be specified, no matter the environment, okay? So this is one example, and the other example is related to gibberellins that are participating in fruit patterning, okay? So gibberellins are important for the development of the valve margin, okay? So this is, so this is the margin between the valves in the Arabidopsis silic, the fruit, and this is super important for fruit opening and seed dispersal, okay? And as you can see here, this is a mutant deficient gibberellin. The valve margin is not well formed, okay? And you can observe a well-formed margin if you treat the plants with gibberellins or if you remove DELA proteins that are the negative regulators genetically. Okay, so in this case, the activity of gibberellins here in the valve margin is super important, okay? And it has to be buffered against environmental changes because the formation of this margin is important for fruit opening and seed dispersal, okay? So I just wanted to point out this before going on. So I close this parenthesis, and as I was mentioning, in many cases, hormones are helping the plant to cope with environment. And in many cases, this environment is a challenging environment, for example, due to abiotic stress. And as you can see here, due to high temperature, freezing temperatures, drought, soil salinity, etc. Okay. And as you know, well, plants are attached to the soil, and what it was a good spot to germinate, it can become a hostile environment later on, okay? And the plant has to cope with this. And in this regard, the hormone abscisic acid, or ABA, it's really important for these responses, okay? ABA is the hormone that orchestrates all the responses that take place in the plant to survive, to tolerate this abiotic stress, okay? So the ABA, well, it's quite well known, and this is the biosynthetic pathway of ABA. The precursors of ABA are carotenoids, and part of the biosynthetic pathway occurs in plastids, as you can see here, and the last part is occurs in the cytosol, okay, in the cytoplasm. And the ABA levels at some point are not only regulated by the biosynthesis, but are also regulated by inactivation pathways. On the one side, there are pathways that what they do is to inactivate irreversibly ABA. For example, just doing this hydroxylation in the position 8. This is a irreversible inactivation of ABA. But in other cases, there are enzymes that what they do is to conjugate ABA to several sugars, and this is a reversible inactivation of ABA. And this is very, very cool because this is a very nice way to regulate the pool of active ABA in the plant, okay? And as you can see here, I have highlighted 3 enzymes in the pathway. These enzymes are catalyzing let's say the limiting steps in the pathway, and they are catalyzing regulatory steps. So among these, NSEED is probably the most relevant because the expression of the genes encoding these enzymes are very, very sensitive to abiotic stresses. For example, the expression of these genes is strongly upregulated in response to drought, okay? Yeah, this is an actual example. No, these are maize plants subjected to drought. And as you can see here, there is a parallel increase in the amount of ABA. You can see here, this is the green line, in parallel with a decrease. Look at the scale in the axis. No, this is actually a decrease in the relative water content in the leaves. Okay, and this increase in ABA probably is due to the upregulation in the expression of the gene encoding the NSEED enzyme. Okay. And what about the signaling pathway? Okay, the ABA signaling pathway is quite well known. It's a relative simple pathway in terms of a number of elements involved. Okay, so we have a positive element. This is the receptor, of course. The receptor is called, well, I mean, in Arabidopsis, these receptors in vascular plants are encoded by small families of genes. Well, not that small. In Arabidopsis, there are, I think, 14 genes coding for the PIL receptors. Okay, this is PIL1, and upon the binding of the ABA, of the hormone, what it does is there is a conformational change that allows the receptor to interact with the protein phosphatases of the PP2C clade. Okay, so these are key proteins in the ABA signaling pathway because they are the negative elements in the pathway. Okay, and what this interaction does is to inactivate the PP2C. So this is quite a common theme in hormone signaling. So there are several hormones, not only ABA, that what the binding of the hormone to the receptor does or triggers is the inactivation or the degradation of the negative element in the pathway. So the pathway, the signaling goes on. Okay, so this is the case. Binding of ABA to the receptor interacts with the PP2C, that is the negative element, and it gets inactivated. Okay, these are only two of the elements. There are more elements in the pathway. Look here, in the absence of ABA, the PP2C, the negative elements are active, and what they are doing is to inactivate another positive element, a kinase called SNERD2. Okay, and the kinase is inactive, and the transcription factors that in the end will change the expression of the target genes are inactive. However, look here, in the presence of ABA, we have seen it binds the receptor, inactivates the PP2C, and then the SNERD2 kinase gets active, and it can phosphorylate the downstream transcription factors that bind to the promoters and activate the expression of the genes. Okay, so among these transcription factors, maybe you know ABI5. So this is probably the most well-known transcription factors in the ABA signaling pathway. Okay, so what are the genes that are expressed in response to ABA? So in response to an abiotic stress, so all sorts of genes involved in, for example, genes encoding enzymes involved in ROS detoxification, for example, or genes encoding enzymes involved in the production of osmolytes, to protect sugars and proline and so on, to protect the cell against osmotic stress, or transporters to move ions or water, etc. Okay, the point here is that, okay, so the expression of these genes and the production of all these enzymes is not enough for the plant to survive to stress. Okay, so the stress pathways have to be integrated with developmental pathways, and this is because when the plant is subjected to stress, this is associated to a growth restraint. Okay, the plant stops growing, because, okay, it's better to stop growing and let's see how to defend, and let's see what happens if this stress goes away. Okay, so this is one of the things that happen. The plants stop growing, and another thing is that the reproductive success is, in some cases, is delayed. In some cases, it's advanced. Okay, so in any case, it's well known that there is an integration between this stress and developmental and growth pathways. Okay, and we have seen ABA, abscisic acid, is the key hormone orchestrating stress, the response of the plant to abiotic stress. Okay, and in the case of growth and developmental program, the key hormone here, I would say, is auxin. Okay, so auxin is a remarkable little molecule that it's able it participates virtually in all growth and developmental pathways in the plant. I mean, indeed, there isn't any known mutant that is able to survive in the absence of auxin. Okay, so auxin is super important for plants. Okay, and well, the auxin biosynthesis pathway is quite well known. I mean, it's quite a straightforward pathway. So the main precursor is tryptophan. There is another pathway starting with indole, but I mean, it's not very well known. The most well-known pathway, and quantitatively the most important, is the one in which the precursor is tryptophan, and it can lead to indole acetic acid, that is the most important auxin in plants, through different pathways. Okay, and they are named based on the one of the intermediates. Okay, and usually these pathways are one or two steps. Okay, so among these, Among these ones, the most important is the IPA. Okay, this is the most important in quantitative terms. Okay, at least in Arabidopsis, that is the plant that in which it's better studied. Okay, so this pathway through the APA pathway is catalyzed by it's a two-step pathway. The first step is catalyzed by an enzyme called TAA, tryptophan aminotransferase. And the second part is catalyzed by flavin monooxygenases that are encoded by the JACA genes. Okay, so just to know. Okay. And similar to what happens with ABA, the levels of auxin at one point is not only dependent on the biosynthetic pathway, it's also dependent on the catabolic and the conjugation pathway. And there has been described several enzymes involved in the inactivation of auxin. No? The main enzymes are encoded by genes called GH3, and what they do is to conjugate IAA to amino acids. Depending on the amino acid, this conjugation is reversible or irreversible. Okay, and there is another way to inactivate IAA, that is methylation by an enzyme called IAA methyltransferase. And this methylation is also reversible, okay, because there are esterases in the cell that remove the methyl group and restore the pool of active IAA, okay? So this is the metabolic pathway for IAA. Another remarkable feature of this hormone is that it can be transported cell to cell through a dedicated transport mechanism, okay? And why do auxin needs a transporter? So this is because of the nature of the indole acetic acid, okay? So as you can see here, the indole acetic acid is a weak acid, okay? So it gets protonated at the low pH that in the symplast, okay, in the extracellular, in the apoplast, sorry, in the extracellular space, around the cell wall, the pH is 5.5, and then the IAA gets protonated and it can diffuse freely into the cell. The thing is that once the IAA is into the cell, the pH in the cytoplasm is neutral. Okay, so it gets deprotonated and cannot exit the cell. And to do so, it needs transporters that assist in this exit. Okay, and there has been described several types of auxin transporters in plants. There are 3 types. One of them is an influx carrier, so auxin can freely diffuse into the cell, but there are transporters that facilitate this. But the most, I think, I would say the most cool ones or the most relevant ones are the efflux carriers, so that are the ones that get the auxin outside the cell, okay? And among these ones, the most important are the pins, okay? And this is because these particular transporters have the ability to be localized in a polar way in the cells. Here in the sample, you can see here these transporters, the pin ones painted in green, so they are located in the bottom of the cell, in 2 adjacent cells, and this is directing or indicating or showing the flow of auxin along these cells. Okay, so this is a very nice way that the plant uses to create, for example, asymmetry in auxin distribution across a particular organ, for example. Okay, so this is another particular feature of auxin that is quite well known. Okay, there is a lot of work done on auxin transport. That is very important also to determine the amount of auxin in a particular part of the plant at a certain time point. Okay, and what about the auxin signaling pathway? So it's quite simple. Also, like as I was telling you for the ABA, in this case, it's very simple in terms of the number of elements involved. Okay. So what we have here in the auxins, we have here 2 elements. The positive elements are the ARF transcription factors, and the negative elements that are the AUX-IAA proteins. Okay, in the absence of auxin, what we have is that the AUX-IAA or IAA proteins bind the ARF transcription factors. What the IAA protein does is to recruit a general core repressor. So the expression of the auxin-regulated genes is silent, okay? But what happens in the presence of auxin? So in the presence of auxin, the auxin binds to the receptor. In this case, the receptor is called tier 1. This is one of the several. There are more parallels in Arabidopsis, but in other plants there is a single one. Okay, so let's say that this is the receptor tier 1, and tier 1, it's interesting because it's a protein with an X box. It has an F box, and it's part of a ubiquitin ligase complex. Okay, so what it does upon binding of the hormone, it's able to interact with the negative elements, the IAA proteins, and send them for degradation. So the ARF transcription factors that were already bounded to the promoters are free to activate transcription, and the expression of the genes go ahead. So this is as I was telling you, this is a quite simple signaling pathway in terms of the number of elements. 2 positive elements, the transcription factor and the receptor, and 1 negative element, the AUX-IAA proteins. And also the theme that I was telling you, the common theme in hormone signaling here, what the hormone does is to trigger the degradation of the negative elements. So this is quite common for several hormone signaling pathways. Okay. So yeah, so regarding the environmental challenges, one niche that provides a continuous challenge for the plant is the underground environment provided by the soil. Okay, and as you can imagine, this is more relevant for roots. So the roots are facing changes in water availability, they are facing changes in nutrient availability, they are all the time subjected to gravity. Gravity is all the time there, is dominant on Earth. So they are all the time responding to gravity, soil type, pathogens, light. Okay, so roots have to respond to changes in all these, let's say, environmental cues. And as I mentioned before, to illustrate, how hormones can mediate the response of the plant to the environment, I have chosen 3 examples that are related with water availability. Okay, that I think is quite relevant for the topic of EpiSeedLink. Okay, and the examples are hydrotropism, that is mediated by ABA. The second one is hydropatterning, that is mediated by auxin, and the third one is xerobranching. Okay, that is mediated by ABA, but it also involves auxin. Okay, so regarding so hydrotropism is, this is quite simple to understand. It's just the orientation of the root growth towards higher water potential within a moisture gradient. Okay, and you can see this here. Here in A, we have a plant growing in a well-watered soil. Okay, so you can see here the root is growing straight down. The root growth is aligned with the gravity vector. Okay, because the roots have positive gravitoprism. Okay, they respond positive to the gravity vector, and they just grow straight down. Okay, but look what happens when this plant is growing in a dry soil. Okay, in which the water source is asymmetrical. Okay, what the root does is to bend, and it's bending and growing towards the water source. Okay, and to do so, the first thing the root has to do is to overcome gravitopism, because gravity is all the time there. Okay, so to understand how plants do hydrotropism, first we have to understand how plants overcome gravitopism. First, we have to understand how plants feel gravitopism. Okay, and this is quite well known. So in the root, there are a special type of cells that in Arabidopsis are located in the columella, that are in the very tip of the root, and these particular cells have a particular type of plastid called amyloplast or statoliths. Okay, and these amyloplasts of statoliths are filled with starch. Okay, so they are heavy, and they are always occupying. They always fall, and they occupy the bottom part of the cells. Okay, look here in this very cool images. You can see here how the statoliths, all of them, are in the very bottom of the cell. So The statoliths are marking what is the bottom of the cell. They are marking the direction of the gravity vector. Okay, and what happens if the orientation of the root changes versus the gravity vector, which is always, it's always the same, of course. You know, and this can happen, for example, if there is a soil movement or something like that. This is an extreme case in which we have moved the root 90 degrees. Okay, and look here how the statoliths that had this position, they fall and they occupy the new bottom part of the cell. So they mark what is again they mark what is the direction of the gravity vector, and this triggers a differential growth response that in the end what it does is to allow the root to bend to align again with the gravity vector. Okay, so what hydrotropism does in response to hydrotropism, what the plant does is to degrade the amyloplasts. Okay, and you can see here in this graph how there is when you put the plant in a soil or in media where there is a gradient in the water availability, the plant starts to bend at the same time or as a consequence of the degradation of the amyloplasts. So as soon as the amyloplast has been degraded, the plants, the roots become less gravitropic and more hydrotropic. Okay, so this allows the plant to forget about the gravity and bend towards the source of water. Okay, and importantly, this degradation of amyloplast also occurs in plants that are in dry soil. And this is important. This is a very nice adaptive response because these plants, they have not subjected to asymmetric water availability. They are in media with very low water available. But what the plant does is to degrade amyloplast just to prepare it in case that there is water from one side or another and this way the plant would be able to move towards the water faster than if they have to start degrading So this degradation in dry soils prepares the roots for possible availability of water in the future. Okay, so where is ABA here? Because I was saying that this is a response that is mediated by ABA. So what ABA does is to promote the degradation of amyloplasts. Okay, so this is one way the ABA is promoting hydrotropism. Okay, and another way it does is to promote the expression of MIS1, that is, well, this is a key regulator of hydrotropism, although it is not well known the molecular function of this protein, because this is this response, the molecular mechanism regulating hydrotropism is not fully known, okay? And the key experiment, the killing experiment here to show that ABA is involved is genetics, okay? And this was shown, of course, using ABA mutants, and this was done in a very nice assay called using split plates. Imagine you prepare a media in a square plate, and when you do regular media, okay, and what you do is to cut in diagonal and remove half of the media, okay, and you fill that half with another media. In this case, media with 0.4 molar sorbitol, or it could be PEG or whatever. The thing is that this is a media in which the availability of water is lower, okay? And what you do is to sow the seeds in parallel to the diagonal and grow the plate, put the plate in a vertical position and grow the seedlings, okay? And as you can see here, this is a wild type. The roots are growing straight down, following the gravity, okay? They are following gravitropism, but as soon as the roots encounter the other media in which the water potential is lower, then the hydrotropism is on and the roots avoid going into that media. Okay, so this is a way to measure hydrotropism. And look what happens with ABA mutants. This quadruple mutant is a quadruple mutant that lacking the 4 PP2C phosphatases that, if you remember, are the negative elements. So these plants are hypersensitive to ABA. And look how the response of these plants is more exaggerated than the wild type. So they are bending much more than the wild type when they are facing this low water potential media. Okay, and the contrary occurs if we use a mutant lacking 6 of the 13 or 14 ABA receptors. So this plant, this sextuple mutant, 1, 2, 3, 4, 5. Yeah, this sextuple mutant is highly insensitive to ABA. And look what happens. These guys go straight into the media. So they are kamikaze. They don't know what they are doing. So the thing is that plants that are not able to sense ABA cannot undergo hydrotropism. Okay, so this is a very, I think this is a quite nice example of a environmental response that is mediated clearly by one hormone, in this case, ABA. Okay, so this is one of the responses, that one of the examples that I wanted to show you. The other one is hydropatterning. Okay, hydropatterning is different, but it's also related to the water, to the availability of water. In this case, what hydropatterning means, this is a term that was coined by the lab of Jose Dineni in Stanford almost 10 years ago. And it says that a non-uniform distribution of available water causes asymmetries in lateral root specification. Okay, so and you can see this very clearly in these examples. Okay, so here there are roots of rice and maize that has been growing on the surface of media, of MS media, or the media that you use to grow maize or whatever. Okay, and as you can see here, the part of the root that is in contact with air doesn't produce, well, a single, but most of the lateral roots are produced in the side of the root that are in contact with the media. Okay, and the same happens with maize. Okay, so this in maize, in this case, the part of the root that is in touch with air is colored with red. Okay, and you can see how all the lateral roots are produced in the other side of the root. Okay, and this is not an artifact due to growing the plants in vitro, because this happens in real life. Okay, and these are X-ray tomographies of maize roots growing in a pot with soil. Okay, and as you can see here, this is in the right panel. You have a root growing in a soil, in a pot full of soil, and you can see lateral roots all over the root. Okay, but look what happens when the root is growing along a macropore of air. Okay, so only lateral roots are produced in the side that is in touch with the wet soil. Okay, so this is the asymmetry. Okay, in the production there is an asymmetry in the production of lateral roots when there is a non-uniform distribution of water, because in this case there is water only in the soil, not in the air, of course. Okay, so lateral roots is a typical response by auxin. Okay, and this is quite well known. So auxin, lateral roots are specified in one particular cell of the pericycle. Okay, the pericycle is an internal tissue that is surrounding the vascular tissue. Okay, so there is a single cell, well, there is a huge accumulation of auxin, and this triggers the production of the lateral roots. And you can see that here. Okay, so this cell file that I'm marking here, this cell file is the pericycle. Okay, and here we are using, well, I mean, the authors were using a reporter for auxin. This is a very popular reporter for auxin, DR5. In this case, it's controlling the expression of GFP, or a Venus, a fluorescent protein, with a nuclear localization signal, so it's easier to visualize it. Okay, so you can see here how, in this case, there are already two cells, but these cells start to divide in. Here we have already two cell files, and in the end, a lateral root emerges. So this is a typical auxin triggered response. Here, auxin is acting as a developmental trigger. A punctual accumulation of auxin in this particular cell triggers the formation of a lateral root. Okay, and these graphs below, you can see how the production is very, very asymmetrical. The blue bar marks the number of lateral roots in the side of the root in contact with media, whereas the yellow one is the part of the root in contact with air. So it's not all or nothing response, but there is a huge asymmetry. Okay, and what this author showed is that hydropatterning is due to differential auxin response in both sides of the root. And you can see that here. In the left panel, you have, this is a transversal cut of the root expressing DR5 gas, in this case. And as you can see here, there is only gas expression in the side of the root that is in contact with, in touch with media, but not in touch with air, okay? And this is what happens in a control root that is growing inside the agar, okay? So the hydropatterning is due to an asymmetrical oxygen response. And how is possible? How can this happen? Okay, so to know how that happens, we have to go to the pathway triggering lateral root formation, and this is the oxygen pathway. This is quite well known, and here I have circled in red the relevant signaling elements for hydropatterning. Okay, one negative element, that is the protein IgG3, and one positive element, that is the transcription factor R7. Okay, so in response to oxygen, IgG3 is removed, R7 is active, and it promotes the expression of downstream genes, for example, the transcription factor LBD16 and other genes, okay? And this leads to the formation of, to the specification of a lateral root and the formation of a lateral root, okay? And in agreement with this, when you count the number of lateral roots in roots that are only one side of the root is in contact with the media, you see that this asymmetry is lost in the R7 mutants. In the R7 mutants, the production of roots in both sides of the root is 50-50, okay? And this defect can be complemented if you transform the mutant with a genomic clone with R7, okay? So perfect, okay? And also in agreement with this, with this transcription factor LBD16 being target of R7, when you look here, these are confocal images of a root where half of the root is in contact with the media, half of the root is in contact with air, and this is a reporter line expressing LBD16 fused to GFP under the control of each the endogenous promoter, okay? And as you can see here, there is more accumulation of the target in the side in contact with media, okay? In touch with water. And this asymmetry doesn't happen in an R7 mutant, okay, so this is fine. But the interesting thing here is that when the authors tested the accumulation of R7 in both sides of the root, what they found, this is a reporter line expressing R7 fused to GFP, what they found is the accumulation of R7 is the same in both sides. So the accumulation of the protein is the same in both sides, but the activity of R7 is higher in the side of the root that is in touch with the media. Okay, so this suggested that maybe the regulation of activity of R7 is due to post-translational modifications. Okay, so what they so when looking at the R7 protein sequence, there are 4 lysines that are susceptible of being sumoylated. Okay, sumoylation is a post-translational modification in which one protein, very small protein called sumo, is attached to a lysine residue, and the sumo is very similar to ubiquitin. Okay, yeah. In fact, the machinery that sumoylates the proteins are quite similar to the machinery that ubiquitinates. But in the case of sumoylation, it usually modifies the activity of the target protein. Okay, so R7 has 4 putative sumoylation sites. What happens when all these 4 lysines are changed by arginines? So the arginine cannot be sumoylated, but the positive charge is maintained. Okay, so the thing is that when the authors did that, a mutant version of R7 where the 4 lysines have been changed to arginines cannot complement the hydropatterning defect of the R7 mutant. However, this mutant version can complement other defects of the R7 mutant, indicating that at least these 4 lysines, these 4 putative sumoylation sites, are super important, specifically for the role of R7 in hydropatterning. Okay, and indeed, what the authors found is that R7 is rapidly sumoylated when you put the plants in air. Okay, so this is a Western blot showing in the middle panel. You can see the amount of R7 and the wild type version of the version with the 4 lysines changed to arginines. And as you can see here, the amount of the protein doesn't change. But when you after putting the plant in air for 20 minutes, the protein is heavily summoylated, okay? And this doesn't happen in the mutant version, okay? And the most important thing is that the authors found that this summoylation, the consequence of the summoylation is that it favors the interaction with the negative element. Remember that I mentioned before that the way the AUX-IAA proteins repress auxin signaling is by interacting with the ARF transcription factors. So in this case, in hydropatterning, I mentioned also that the negative protein involved this IAA3. So what the author found is that this is an in vivo pull down. And look here in this panel, and you can see here is that the wild-type version of ARF7 is able to interact more strongly with IAA3 than the version in which the 4 lysines are changed to arginine. So this result suggested the authors that summoylation favors the interaction with the negative elements, okay? And the summoylation happens in air. So in the end, the model that the authors proposed to explain how plant undergoes hydropatterning is that is due to a differential, to a symmetrical summoylation of one key protein in this pathway, that is R7. So R7 only gets summoylated in the side of the root that is in touch with air. The summoylation favors the interaction with the negative element. So R7 is inactive and it cannot promote the formation of lateral roots. However, this negative regulation doesn't occur in the side of the root that is in touch with water. Okay, and in this case, lateral roots are formed normally. Okay, so I think this is a quite very cool example of how, in this case, auxin is mediating the response of the root to one environmental cue, that is the asymmetrical distribution of water. Okay. And the last example is a more extreme situation than this one, okay? And it's called xerobranching. And xerobranching occurs when the root loses contact with the soil, okay? So when the root enters a macropore and loses contact. In the case of hydropatterning, one side of the root was in touch with the soil. In this case, the root has lost contact with the soil, okay? And this is an adaptive response that what it does is to suppress totally lateral root formation, okay? And this response is mediated by ABA, and it also involves auxin, although the leading role here is done by ABA, okay? So here it was shown at the beginning that this was an ABA response. Here, this is the experimental setup. Okay, so you have here, plant is growing here. These are the roots. And when the roots enter the macropore, there are no lateral roots from here. These are roots of tomato, okay? And these are tomato mutants, notably Sunflaca, that are ABA deficient. These mutants cannot produce ABA, okay? And look, they cannot produce ABA, and they are forming lateral roots here. And these plants, they don't have any need to produce lateral roots here, because this is a waste of energy, but they are producing. So this experiment, they showed this with tomato and Arabidopsis and several other species. So this is a very well conserved response, okay? So the ABA mutants have altered xerobranching. They cannot undergo xerobranching, okay? So the authors, well, this is a work done in the lab of Malcolm Bennett in University of Nottingham. He has been working a lot with the plant response to water, okay? And they wonder where in the root ABA synthesis occurs to regulate xerobranching, okay? So what they did was a very nice experiment. This was done in Arabidopsis. This is the same setup. Here, instead of soil, is regular media. You can see the root growing through the media, and the root goes into the air gap, and there are no lateral roots formed. And then the root enters again the media, and lateral roots are formed again. Okay, but look what happens in the ABA2 mutant. That is an ABA deficient mutant. Lateral roots are formed even in the air gap. Okay, so what the authors did was to try to see where in the root ABA synthesis was important, and what they found is that synthesizing ABA just in the flowing companion cells using the SAC2 promoter. So production of ABA just in the flowing companion cells is enough to complement the serobranching defect of the ABA2 mutant. Okay, so from this experiment, they can conclude that the ABA produced in the flowing companion cells is required to suppress lateral root formation. Okay, and this result is important if we put in the context of other results that were already known, for which there was experimental evidence and also evidence from mathematical modeling, and it's shown here. Okay, so here what this says is that when the root is in a well-watered soil, there is a dominant inward water flow. Okay, this is already known. Okay, so the plant takes the water from the soil, entering through the epidermis, and the flow is inward towards the vasculature to transport the water to other parts of the plant. However, what happens when the root loses contact with the soil is that this flow of water is reversed. Okay, so when the root has lost the contact with the soil, the water that in the root comes from other part of the plants through the phloem. Okay, so there is an outward water flow. Okay, and what the authors thought is that, okay, so this outward water flow coming from the phloem might transport the ABA produced in the phloem to other parts of the plant. Okay, and indeed, this is what they found. So these are roots of Arabidopsis expressing a ABA sensor called abacus, and what you are imagining here is mainly the epidermis. Okay, and the whiter, the more ABA. Okay. This is a direct sensor. It's measuring directly ABA, so you can quantify it. Okay, and as you can see here, as soon as the root enters loses contact with the soil, there is a higher accumulation. There is a progressive accumulation of ABA in the epidermis. So this ABA is coming from the phloem. Okay, and importantly, this is a transient response, because as soon as the root enters in the media again, this ABA in the epidermis disappears. So this is a transient response. So ABA produced in the phloem companion cells reaches the epidermis when the root has lost contact with the soil. Okay, so this was very nice. Okay, and how this is related with the production of lateral roots. Lateral root production, as we have seen, is an auxin response. Okay, so what the authors did was to check in the same experimental setup, they check the amount of auxin. Okay, so using a reporter, in this case is DR5 expressing Venus with a nuclear localization signal. Okay, and in this part of the root, auxins are moving through the epidermis. Okay, and from there they move inside the cell. Okay, and as you can see here, the amount of auxin in the epidermis, because what we are looking here is at the epidermis, is increasing as the root enters in the air gap. Okay, and again, and similar to what happens with ABA, this is a transient response, because when the root tip enters in media again, the amount of auxin in the epidermis is reduced. Okay, so there is a transient increase of auxin in the epidermis. It seems that the auxin is trapped in the epidermis, because, as I mentioned, auxin is moving through the epidermis, but it cannot move inside the root when the root has lost contact with the soil. Okay, and how this is related with what we have seen with ABA. So here, well, these guys did many more experiments, but the killing experiment was showing that what ABA does is to close the plasmodesmata. Okay, plasmodesmata are contacts, let's say holes in the cells that put adjacent cells in contact. Indeed, all the cytoplasm of adjacent cells that are connected by plasmodesmata are in direct contact. So, and this is called the symplast. Okay. So what the authors found is that ABA closes plasmodesmata, and they showed this using a fluorophore called CFDA, this CFDA is uptaken by the cell, and once inside the cell, it is cut, and then the CF part gets fluorescent, okay? And it only moves from one cell to another through plasmodesmata, okay? And as you can see here in these pictures, you can see, well, a wild type and an ABA insensitive mutant. In regular media, the CF, the fluorophore is all over the root. But look what happens when the root loses contact with soil. The fluorophore is restricted, is trapped in the epidermis, while it's all over the root in a plant that is insensitive to ABA. Okay, so in a plant that is insensitive to ABA, plasmodesmata are open, while in a wild-type plant, when it loses contact with soil, plasmodesmata are closed, and the fluorophore, the same like auxins, are trapped in the epidermis. Okay, so all these experiments allow the authors to propose this model that is very nice. And so this is the situation. Okay, so this would be this marks the different tissues in the root, and this would be a root growing in a well-watered soil or in a regular media. And what we have is that, I mean, this is water. This is the flow of water. There is an dominant inward flow, water flow, because there is water in the media, and auxins that comes through the epidermis are able to move inside to the inner tissues. Among them is the pericycle, where the auxins can trigger the formation of lateral roots. However, look what happens when the root loses contact with soil. When root loses contact with soil, ABA is produced in the phloem, okay? And the water flow is reversed. Then the water is not coming from the soil because there is no water in the soil. The root is in air, but the water is coming through the phloem, and then the phloem moves, the ABA moves through across the root with the water flow, okay? And with time, the amount of ABA is higher, and this ABA triggers the closure of the plasmodesmata. And what happens with auxin? Gets trapped in the epidermis. It cannot reach the pericycle, and it cannot trigger the formation of lateral roots. Okay, so this is the mechanism that was proposed by these authors to explain this very cool response that is called xerobranching. And I think this is a very nice adaptive response that the plants have to cope with this sort of soils that probably this is more and more, this response is more and more relevant when in dry soils, no, because it's more easy for the root to find big holes in the soil where it loses contact with the soil. Okay, so this is what I wanted to tell you. So I would be happy if you by now should know that hormones mediate many responses to the environment. Okay, I have mentioned just 3 of them. Know that in some cases this is important. Not always the hormones are responding to the environment. In some particular cases, the hormone activity is buffer against environmental changes because they are integral parts of the developmental program that have to occur no matter the environment. Okay, and you should know, or at least understand the basics of the ABA and auxin pathways, and in the end be able to list at least 3 responses of roots to the availability of water and what hormones are involved. Okay, and this is all I wanted to tell you. So if you have questions, I will try to answer them. No questions? At least I hope you liked it.
Hello, I have one question.
Okay, so go ahead.
Okay, so it was super interesting, but I didn't get very well the relationship between the auxin and the ABA in the long term. Yeah.
So what ABA does is to look in when the root goes loses contact with soil, Okay, so ABA is produced in the phloem.
Yeah.
You know, because ABA is feeling drought. The plant is feeling drought, and it induces the production of ABA. And in this situation, the flow of water is reversed because there is no water in the media because the root is in air. So the water comes from the phloem and it takes the ABA produced there and it moves across the root. What the ABA does is to close stomata.
Yeah.
This indirectly causes the accumulation of auxin.
Okay.
The auxin gets trapped in the epidermis. There is not a direct relationship between ABA and auxin. The effect is indirect. So what the ABA does is to close plasmodesmata. Closes plasmodesmata. And then the auxin cannot move inside the root.
Okay,
And
it's just that it allows the movement.
Yeah.
Yeah.
Yes,
Okay.
yes. There is no ABA.
Okay.
Plasmodesmata is open. And usually the flow of water would be inward and then it moves the auxin through the plasmodesmata across the root towards the inner issues.
Okay, perfect. Thanks.
Sorry, I also have a question about the hydrotropism, about the statolis. I mean, the statolis. In your slide, you said when suffer from the water deficit, the statolis will digest it, right? But I will question about is the because of digestion or when the plants lack of the water, they will not able to produce more those statolis. It's because of digestion or the ability of producing this stuff.
Yeah, I don't know. The amount of statolis is lower. So, and you see a reduction in the amount of statolis. They are probably, I don't know what is the mechanism that the plant uses to degrade it. It's induced by ABA, this is for sure. ABA induces the degradation of statolis, or amyloplasts is the other name used for these organelles. And I cannot tell you the details of this because I don't know.
Because there are different mechanisms if the digestion and the
It could be, it could be an autophagy mechanism involved autophagy, for example, that is removing the whole organelle, you know, or maybe it's just a degradation of the starch inside the organelles. So, because what it makes the statholids or the amyloplast heavy is the amount of starch that is inside, you know, because they accumulate lots of starch. Maybe the plant, what it does in response to drought, or maybe in response to an asymmetric distribution of water, is to degrade the starch inside the statolids. I'm not sure about that. Or maybe it's the whole statolids what is degraded. I cannot answer that.
Yeah.
But probably if you are curious about that, checking the literature, I'm sure there may be something about that.
Okay. Thanks.
Hello. I have a question. Not particularly related to the major topic which you discussed. Thank you for the presentation, though. If at all, per se, in a situation which I want to consider, that we have a compound which interacts with the seeds in the germination perspective, the compound which interacts with the seeds and which increases the gibberellic acid pathways, like it makes the ratio of gibberellic acid more than the ABA, so that the germination rate increases. So per se, if the germination was supposed to happen on 3-day basis, third day, the germination happens on the second day itself. So if this is the situation, if this happens particularly with particular compound, will this pattern be followed throughout the life cycle of the plant? Will the growth pattern be the same? Will it be increasing always?
What you say is that if the ratio between gibberellins and ABA induced by the compound will be maintained, I don't know. We should know what is the mechanism used by the compound to alter the ratio of GAs and ABA to try to understand whether this can be happening all over. But what is your concern that maybe this compound is making the plant less responsive to stress or something, for example, or?
Yeah. So yeah, my concern was the same in the sense like if at all this particular compound increases the gibberellic acid content and allows the plant seed to germinate faster, even in any of the given circumstances, like if there is a drought condition, but still the seed germinates quicker.
Yeah, but you know, there will be, I mean, in drought, what we know for sure is that there is a decrease in gibberellin synthesis, endogenous gibberellin synthesis. So there will be here a contrasting effect. So for maybe the compound is triggering GA biosynthesis, drought is triggering reduction in GA biosynthesis.
Right. Yeah.
I don't know. This is something that you should check. I mean, and we could check that also.
Yes.
If we have your compound.
Yeah. Yeah. Thank you so much for that.
Hello.
Hi.
Hi. So I have a question regarding the seed germination. So as you explained that the seed germination would be delayed when there is ABI5, if I'm not wrong, like they inhibit the seed germination, right?
ABI5, the transcription factor, you mean?
Yeah.
Yeah, I guess. No, I mean, it's induced by, it's activated by ABA, it should delay germination. Yeah.
Okay, and I also know that that the DELA proteins, which you were like initially talking about, they somehow inhibit the GA signaling pathway. So is there an interaction between both of them, like in mediating the seed germination, that when there is ABI5 higher in a plant, in a seed, and then the DELA, they have somehow crosstalk with each other, and then they...
I mean, I cannot tell you the mechanism, but I'm sure that there is some crosstalk because, I mean, DELAs are, I don't know if maybe someone has published that DELAs are interacting with ABA5. I mean, we have found interaction with many transcription factors, and some of them are BCIPs, like ABA5. So I wouldn't be surprised that DELAs are, and checking the literature, I'm sure that someone has published, and in this case, DELAs and ABA5 both are repressing the germination. So they will be playing together for sure. So this antagonism between ABA and gibberellins can be explained because gibberellins promote the degradation of DELA proteins. So if DELAs are required for high-5 to repress germination, so this could explain, not because gibberellins are removing DELAs and high-5 is not, ABA5 is not so active. So checking the literature, but I'm sure that there is someone has shown that they are interacting for sure.
Sure, I will check that, and thank you.
Thank you very much for your lecture. So I have a question still regarding the statoliths. So you said like when there is uneven distribution of water, the statoliths are degraded to overcome gravitropism. So what happened in a case where
it's not even distribution of, there is even distribution. The thing is that is when the plant is facing drought. So there is no water at all, or very little water.
Okay.
So the plant degrades the statoliths to be prepared to do hydrotropism, assume that there is water.
Okay. So now when there is water again, drought,
Yes.
like when there's water again, let's say towards gravity, is the statoliths reproduced, or that's like permanent degradation?
No, I mean, I cannot tell you this for sure because I haven't seen any experimental evidence about this, but I'm sure that there is some published. But I would bet that this is not irreversible. This would be bad for the plant. The plant doesn't want that. I mean, the plant needs to, this is an adaptive response to be prepared when the plant is facing drought. But if suddenly, I mean, there is rain and the drought is gone, the root wants to be responsive to gravity. So I'm sure that the statoliths, the amount of starch and the number of statoliths is increased again, for sure. This is, I mean, it would be very reasonable that this is, I mean, many of the responses that the plant does regarding the environment are reversible. I mean, because the environment is changing. So for the plant, doesn't want to take irreversible decisions. So in many cases.
Thank you.